111. This is going to be a lab on DNA. Where's my thing? On DNA extraction. And then there's a CSI. There's going to be two whiteboard videos on this, one to explain the DNA extraction and the CSI. Also, if you look in the module, I found a video on how to actually do this lab. So you could actually do this lab. You could do this lab at home if you want, using household chemicals. It's really kind of cool, and if you have some little kids around, they might really enjoy sharing the experience with you. So click on that video and you'll see I wrote a thing in there. So let me talk about DNA. I'm going to do this a little bit differently because I'm going to refer to the actual experiment itself. You have a copy, so you can go through this. So let me explain DNA. So DNA stands for deoxy, big words, ribonucleic acid. And this is the genetic material. This is the blueprint to make a whole new you, or a whole new organism. All right, and DNA basically has all the information to make a new human, and it basically does it in 3-letter words from a 4-letter alphabet. And many of you, I'm sure, have had some biology in high school, but I'm going to go through this very briefly. And if you look at the lab manual, it'll talk about it. So the 3-letter words are what are called codons. And the 4-letter alphabet, the 4 letters are called nucleotides. And these are molecules. The basic idea of the way the information is transferred is DNA, before any cell divides, or even when you're conceived and there's a zygote, you start with 1 cell that becomes 2, 4, 8, 16, and then DNA makes copies of itself. This process is called replication. All right, and remember, so every time a cell divides, it makes a copy of DNA. The DNA then, if it's going to actually make an organism, it makes a working copy. So if you think in terms of a building, like the buildings at Mesa College, in an office somewhere in Sacramento, there are the actual blueprints to build every building. But when they actually build the building, they may have a working copy of that blueprint. That's what the contractors work on. That, in this case, is an RNA copy, and RNA is ribonucleic acid. This process is called transcription. So DNA makes an RNA copy. And then the working copy is the information to make protein. So DNA does not make RNA, and RNA does not make protein. DNA contains the information to make RNA. RNA has the instructions to make protein. This process, by the way, is called translation. Now, how does DNA make a copy of itself? This is where you can look at the lab manual here. So DNA, first of all, it does have a direction, just like reading anything. There's the beginning of the sentence and the end of the sentence. They're labeled 3' and 5', and DNA is self-complementary. So the 4 letters are, if we go back to here, the 4 letters are, that 4-letter alphabet are T, A, G, and C, and those are just abbreviations for the nucleotides. It turns out on a piece of DNA, DNA has 2 strands. They are anti-parallel, meaning that the 3' side here will have the 5' side here. But you'll notice if you look at this diagram here, that every time there's a T on one side, there's an A, and every time there's an A, there's a T. So if you know the sequence here, you can figure out the sequence there. If there's a C, there's a G. So the complementarity is that strand 1, strand 2, if this is a C, this is a G. If this is an A, this is a T. And so if you follow along here, if I tell you here's the DNA strand, T, A, C, C, whatever, you can figure out what the other DNA strand would be. And when you cut these in half, each of these has the information to make another copy, and that's how the replication works. You cut this in half, and then you can use this to make one of these, and you can use this strand to make one of these. Now, transcription works similarly in that what happens is DNA, one of these two strands has the actual information, and usually it's this one. All right, this is just a template. Then what happens is DNA leaves the nucleus of the cell, and it can make an RNA copy. And there's several differences between DNA and RNA, but in RNA, there are no T's. Instead, there are U's, and U stands for uracil. And why that is, it kind of just worked out that way. But if you take the DNA sequence here, right, T makes an A, see how the RNA, T makes an A, but see where A makes a T, if it's RNA, it'll make a U. And so then we have, from the DNA strand, you can figure out what the RNA strand. And again, remember that the 3' end of DNA will make a 5'. They are what's called anti-parallel, which is head to tail. All right. Once once we make the RNA, we translate it to protein. We translate it to protein using what's called the genetic code. And what this does is it decodes the 3-letter words. And so you look at the 3 letters, and you start in 3s, and then the first letter is U. This is the way you read this. Is you have U is the first letter, then the second letter is U. You would look in this box, and then the third letter, right? So if it's U, 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 then it's going to be F, which stands for phenylalanine. If it's A, C, A would be a T here. See that? So for example, if you follow through here, the genetic code's here, and I'll just kind of put this over here so you can see it better. All right. If I have the RNA, AUG, and the other thing is not everything gets translated, so there is a start here, just like a capital letter and a period. All right, so the first thing you do on a piece of RNA is you look for where it says AUG. AUG means start, and then you can literally do this. You say AUG is start, so if you say AU is this box, G is start. If AUG is in the middle, it's an M. But once you go that, then we start doing this. We go U, G, G, and that stands for W. Then AU is this box. AUC, right, is an I. AA is this box. AAU is N. And then GAG would be in this box, and GAG would be the E. And then there are some that just mean stop, just like putting a period. Right? And so you see how this actually spells out wine. I just make it spell a word. Interestingly, mutations are changes in the DNA, and you can see if you follow through this, it goes through a whole bunch of stuff about mutations, how you can have a mutation that changes a letter, and the lab manual explains a lot of that stuff, but you can play with that a little bit. Right? Now, the video shows you how to isolate DNA from strawberries. DNA is water soluble. It's not soluble in alcohol. And because DNA lives inside of cells, inside of the organism, the cells have to be broken open, so we use a little bit of detergent to do that. And then you add some alcohol, and the DNA is insoluble when it comes out. And if you watch the video, it's actually really cool. Right? I don't want to go through a whole lesson on genetics, but that's basically how DNA works and what it does and how we get protein from it. Right? That should help you answer a lot of the questions at the end of the lab. I'm going to do a second video on the CSI part because it's also very interesting and I think you guys will like it.